Hi, everyone. Welcome. And welcome to the Zoom audience too. I think you're connected into this room as well. Welcome to tonight's Research Spotlight talk at the Yale Ph.D. Museum. This is a graduate research speaker series that we have been doing for the past 2 years. And it's a wonderful time to highlight the work that different graduate students, different research that graduate students do in connection to our museum's collections. So welcome. And I'm Katrina Damrell. I'm a Master's of Environmental Management student at the Yale School of the Environment. And I'm also the Research Spotlight Assistant working with David Heiser, who is off screen here, but also managing the Zoom Q&A, which I'll get to in a second. Actually get to it right now because I already introduced the speaker series. So as you guys know, the Peabody Museum has also been in the process of reopening, and we're excited to be in this space. It's called David Friend Hall. And as you can see, it's a pretty special area to be in. And it's the first time that the public has been invited into the space. So congrats on being some of the first people to see this. And for everyone on the Zoom world, please stop by soon. A few housekeeping details. So as you know, it's a hybrid event. So for everybody in Zoom, you can post your questions at any point during the talk and we'll capture them. David's off screen here. He's monitoring that and it won't throw us off at all. But if you have questions in the audience here, hold on to them until the end and we'll do a Q&A and we'll toggle between the in-person and Zoom format so everybody can get their questions answered. That said, I will also say we usually run out of time. So if you do have more questions, Caleb here, who I'll introduce in a second, is happy to answer any questions directly. And we'll share his contact information. And then we're also being captioned. So if you're in the Zoom verse right now and want to use live captioning, Becky is helping us tonight. Thank you, Becky. So you can turn your live captioning on, and I'm looking upwards because the camera's up here if anyone's confused. But feel free to turn the live captioning on. It's on the bottom right corner of your screen and on your Zoom screen. Okay, I think that covers everything. And we will be passing around mics too. So anybody who has questions here afterwards, the audience in Zoom can also hear. Today we're joined here with Caleb Gordon. Caleb is a PhD candidate at Yale's Department of Earth and Planetary Sciences. He grew up in Brooklyn and went to Bowdoin College in Maine. And his work focuses on how reptiles evolved to become the top predators of the food chain on land and in water during the time of the dinosaurs. And today he'll show us how these ancient marine reptiles have evolved using a combination of classic paleontology methods, World War II naval statistics, and machine learning, all combining to solve some of the most peculiar puzzles known in evolutionary science. So without further ado, thank you.
Hello. Testing the, thank you all so much. Testing the microphone. Can you all hear me okay? Okay, fantastic. Thank you so much for that wonderful introduction, Katrina. And hello, everyone. It is such an honor to present in this space. This place is beautiful. Thank you all so much for joining me today. I really, truly do appreciate it. And as Katrina said, my name is Caleb. I'm a PhD student here, and I study marine reptiles, creatures that are often overshadowed in paleontology because most of them lived during the Mesozoic era, which we tend to think of as the time of the dinosaurs. But while dinosaurs walked the earth and pterosaurs filled the skies, giant marine reptiles ruled the world's oceans. And these animals were just as diverse and epic and remarkable as the dinosaurs that walked on land above them. These marine reptiles include the better known plesiosaurs, ichthyosaurs, and mosasaurs, but also a host of other lesser known creatures more or less distantly related to them. And one of the fundamental questions we have in studying these animals is when and how they first adapted to life in the water. Because all reptiles descend from creatures that lived originally on land. Our most distant fish-like ancestors evolved to leave the water long ago, when one group of them evolved special land-proof eggs that allowed them to voyage inland. Reptiles were part of this group. Their ancestors lived on land. But then, over the course of their history, dozens of groups of reptiles independently returned to the water. None of these groups were ancestrally or primarily aquatic. Instead, we call them secondarily aquatic because they adapted to life on land first and life in the water second. We can follow this transition in the fossil record by looking at the limbs of these species, which hold a lot of clues for how they first adapted to life in the water. Marine reptile limbs follow a common trajectory. Most land-dwelling reptiles have unwebbed hands, with space between the fingers, like our hands have. But when they go in the water, their hands become more webbed over time, until, in the most aquatic species, their hands evolve into flippers. We expect these changes to be reflected in the structure of the limb skeleton. For example, some studies have suggested that the overall shape of the hand skeleton might change over time to reflect how webbed it is. In addition, the bone at the tip of each finger, the one that bears a fingernail or claw, might get flatter and longer over time to support more webbing. For the same reason, the bone at the base of the finger might get wider over time to help suspend the larger webbing surface. And finally, paleontologists have suggested for a while that the hand might get relatively longer and the rest of the arm relatively shorter as a group becomes more aquatic over time. Each of these features is like a different breadcrumb trail that we can use to follow their evolutionary journeys across time. And yet, no matter how, forgot to mention this. So the hands will get relatively longer and the lower arms might get relatively shorter. And this is another one of the features that's been suggested. And we have so much potential data from all these features, but I would venture to say that at the state of the field where it is right now, no matter how much data we collected on all these features, somehow paleontologists would manage to disagree about exactly how aquatic all these creatures were and how these features evolved. And that's because there's a critical and underappreciated issue for marine reptiles in particular. Today, we're going to talk about why that is. What makes marine reptiles so difficult for paleontologists, and why can't we all just finally agree? We'll discuss one emerging way to solve this problem, and then we'll see what our new solution tells us. So to begin, what makes aquatic reptiles so tough to study? What makes them so darn difficult? Well, the problem ultimately comes from the fact that being aquatic is a spectrum, and it's hard to reliably put extinct species on this spectrum. Even today, there are modern animals on every point of this spectrum. Some species are fully terrestrial. They will never voluntarily swim. Others are slightly or transiently aquatic. They go in the water occasionally, but usually they're on land. Others are moderately aquatic or amphibious. They spend a lot of time both on land and in the water. Others are highly aquatic. They spend most of their time in the water, but still occasionally go onto land. And then others are fully aquatic. They never intentionally leave the water. For each of the major groups of marine reptiles, we want to trace their evolutionary history through these hypothetical stages so that we can tell the story of how and why they adapted to life in the water in the first place. But for species in this semi-aquatic range, it can be really challenging to stage them correctly because different features in their skeleton will say different things about just how aquatic they were. For example, a crocodile may have paddle-shaped hands that help it swim better in the water, while retaining tall, sharp claws that help it dig well on land. Species like this spend some amount of time both on land and in the water, so they have multiple different features that have adapted to each of these environments independently. Now, some extinct species like ichthyosaurs and plesiosaurs avoid this problem. All of their features agree in suggesting that they lived exclusively in the water, and no one really disagrees that they were fully aquatic. But others, like the older cousins of plesiosaurs, a few marine lizard relatives, several early reptiles, and some aquatic dinosaurs have been much harder to place on this spectrum. because they weren't quite fully adapted to life in the water, so they have a blend of aquatic and terrestrial features. And for many of these species, different features say conflicting things, leading to scientific disagreement. A contentious fossil will have some features that lend themselves to a terrestrial interpretation, and others that lend themselves to an aquatic one. And this conflicting evidence can spark some pretty intense academic debates about the aquatic habits of extinct species. Cases like this often trap us in a stalemate, where different researchers will stick by their favorite line of evidence without coming to a resolution. This is one of the fundamental problems we face when trying to determine just how aquatic these extinct species were. Different lines of evidence often say different things. So how can we solve this fundamental problem? How can we choose between conflicting lines of evidence in paleontology when they disagree? Well, as it turns out, we can combine two approaches, one kind of new and one kind of old, that let us choose the best line of evidence to predict aquatic habits in 2 steps. For our first step, we can use a subject that's all the rage right now, machine learning. This is just any technique that learns about known things to predict unknown things. In our case, we can take modern species whose lifestyles we know, and we can use measurements collected from them to train a machine learning model. And then we can repeat this process, testing and retraining our model again and again and again until the model is really smart and has learned a lot. Then we can use this smart model to predict the lifestyle of a given extinct species. And we can do this once for the shape of the hand, once for the shape of all the finger bones, and once for the relative lengths of the hand and the lower arm. To see how these models work, we can also look under the hood at what they're actually doing. Each model stores measurements of some feature, say relative hand length on the x-axis here, for modern terrestrial species and modern aquatic species, and fits as best as it can a squiggle that can predict the probability that a given species is aquatic based on those measurements. So then, if we measure the relative hand length of some mystery fossil, our model will tell us the probability that it's aquatic. Using this approach, we can make predictions in the same way for every limb feature that we want to study. But we still need a way to pick the best results. And fortunately, the US Navy happened to already have invented one for us. Because during the Second World War, US aircraft carriers used radar to detect incoming planes. People operating the radar measured, whoop, seems to be an alarm. Oh, all good. Okay, nice. This is high stakes. That's all part of the 4D experience. This is actually the perfect slide for that. So during the Second World War, US aircraft carriers used radar to detect incoming planes. The people operating those radars, radar operator receivers, they were called, measured different characteristics of the signals they received on the radar on screens like that, which they used to make models that could predict a plane's allegiance, whether it was an ally plane or an Axis plane. And sometimes when they saw a scary, mysterious incoming plane, different models might disagree about whether it was friendly, which could lead to a game of high-stakes Dora. What should we do? To pick between these conflicting models, the US Navy develops what we now call receiver operating characteristic, or ROC curves. These were just a quick way to compare how accurate different models were. ROC analysis uses 2 measures of accuracy: how many true positives you have and how many false positives you have. Now, these terms are jargony, but you might be most familiar with them from the pandemic. A true positive is anything that's correctly identified as belonging to a group. So if you are really sick with COVID and you take a COVID test and test positive for COVID, that's a true positive. You are truly positive for COVID. A false positive is anything that is incorrectly assigned to a group. So if you do not have COVID and you're not sick at all, and for whatever reason you just enjoy taking COVID tests, which would be a little weird, and you take it and you test positive for COVID, that's a false positive. You are falsely positive for COVID. So the US Navy cared about these 2 metrics because they naturally wanted to avoid intercepting friendly planes and ignoring dangerous planes. So to do that, they needed models with the fewest false positives and the most true positives. And they did that by fitting those models on a curve like this, where the percentage of true and false positives became the axes. Now, anything to the far left on this curve, I think that's left for you guys, is ultimately just describing a more accurate model, and everything down and to the right is describing a less accurate model. And so what the Navy would do to use this method to determine which of conflicting results they would take is they would connect the dots. They'd look at how accurate each of those models was in different campaigns, and then they'd connect the dots for each model to create ROC curves describing overall model accuracy. And then they'd pick the model farther up left to make their decision. Problem solved. This technique has been used for decades in medical work, in military work, but it's hardly ever been used in paleontology, even though it's kind of exactly what we need, a way to consistently pick the most accurate machine learning model to predict aquatic habits. Because remembering our problem from before, secondarily aquatic groups can be tough to study when they have different features that say different things about just how aquatic they were. We've now found an interesting solution to this problem, one that combines modern machine learning with an old military-grade approach to picking the best of many models. With this toolkit in place, we can explore what our solution actually tells us by collecting the data on all the limb features I mentioned before, running it through our machine learning approach, and seeing what that approach shows us about the aquatic habits of contentious fossils. Collecting this dataset involved a bunch of field and wet lab observations to record the aquatic habits and limb phenotypes of modern species, mammals and reptiles alike. It involved also a lot of specimen photography and CT scanning of specimens preserved in jars, where I'd take a specimen like this gharial here from India, put it in front of a CT scanner, and scan it so that I could get a 3D digital version of the skeleton to measure its limb bones. By CT scanning this entire croc, we can digitally dissect each layer of the specimen without damaging it to get access to its entire skeleton. Then everything I photographed and CT scanned had to be measured. I measured the limb bones of many flattened skeletons using 2D image annotation software, which should appear here. And I measured the limb bones of scanned specimens using 3D mesh manipulation software. These approaches allowed me to amass hundreds of extra specimens from around the world that I wouldn't have otherwise been able to sample from countries that I've never been to. But for the majority of specimens in my dataset, I measured things the old-fashioned way, going through drawer after drawer after drawer of modern and fossil specimens, most of which were here in the Peabody right below your feet. And for those on Zoom, hopefully they are not below your feet right now, or that would be a felony. And measuring them to the nearest millimeter, all of these specimens with calipers. Now, using this blend of physical and digital measurements, I collected a series of measurements for each specimen that could capture the shape of the finger bones, the shape of the hand, and the relative length of the main limb regions. In doing so, I compiled the largest data set of vertebrate limb measurements to date, now including over 750 specimens and almost 12,000 measurements. And this gave me enough data to train and test machine learning models to predict aquatic habits using each of the major limb features I mentioned before. Ultimately, these models showed that some limb features were better than others. So not all limb features are created equal when it comes to predicting aquatic habits. And as a refresher for this, because we're about to look at some ROC curves, just remember that I'm comparing these models within an ROC space where if a curve is closer to the top left-hand corner, it's more accurate. It's better at making predictions. And if the curve is farther down here, then it's less accurate. It's worse at making predictions. And when we plot our 4 new models in ROC space, we find that there's a clear winner. Relative hand length, shown in green here, is far and away the best predictor of aquatic habits. What this graph implies is that by just knowing the relative lengths of the hand and the lower arm of a species, you can predict how aquatic it is over 90% of the time across mammals and reptiles. That is incredible because it gives us an unprecedented ability to determine the aquatic habits of a new species just by looking at the bones in its arm. If we compare this model to another one that used finger bone shape instead, we see that that model kind of stinks. It doesn't predict aquatic habits any better than you could by just closing your eyes and guessing about them. To give you a feel for why these models perform so differently, we can look at the actual data that these two models were using to be trained. These graphs show what the majority of values look like for each of our lifestyle bins, from fully terrestrial to fully aquatic. And on the graph on the left here shows relative hand length. So as you go up the y-axis, the hand is getting longer relative to the rest of the arm. And there's a clear directional trend from fully terrestrial to fully aquatic upwards. So what we see is that there's a clear trend to get longer hands as you become more aquatic. Whereas if we compare this to first finger bone shape, as the width of the first finger bone changes, there's no clear directional trend from fully terrestrial to fully aquatic. In other words, fully terrestrial and highly aquatic species have very different limb proportions, but don't have very different, on average, finger bone shapes. So this explains why limb proportions, the relative length of the hand, is really good at predicting aquatic habits, but finger bone shape is not. We've come a long way now from where we started. In studying ancient marine reptiles, we came against this problem that different lines of evidence tend to disagree about how aquatic they are. And to address this problem, we found a solution that combined machine learning and ROC analysis. By applying this solution, we found that all of the limb features we studied, of all of them, the relative length of the hand made by far the best predictions about the aquatic habits of extinct species, which is fun because that's actually like one of the easiest sets of data to measure of all the things we looked at. Now, to wrap up, let's take a preliminary look at what this best model actually tells us. It's right 90% of the time. What are its predictions about the aquatic habits of these extinct species? Well, so far I've used this hand length model to determine the aquatic habits of just a small subset of contentious species within the dataset. This is a plot of predicted probability from 0 to 1 that a given species is highly or fully aquatic. So a 1 means that without a doubt, it's highly or fully aquatic, and a 0 means that without a doubt, it's frequently coming onto land. And just to help orient us, shaded in red here are the values we get, sort of the predicted probabilities when we just happen to feed all of the modern not highly or fully aquatic taxa into the model. And in blue here is when we feed all of the modern highly and fully aquatic taxa into the model. And what we find is that when we feed contentious extinct species into this model, the earliest ever semi-aquatic reptiles like Mesosaurus and Spinoequalis, as well as many plesiosaur relatives like pachypleurosaurs and nothosaurs, have an amphibious lifestyle, regularly returning to land. Unsurprisingly, our model also recovered clear highly or fully aquatic habits for ichthyosaurs, plesiosaurs, geosaurs, and mosasaurs. But more contentiously, it also recovered highly aquatic habits for some lizard relatives called pleurosaurs and for Spinosaurus. Now, the ecology of Spinosaurus in particular continues to fuel intense debate. Frankly, I think Spinosaurus might be in one of that 10% false positives here, but stay tuned for the next round of our analysis for the whereabouts of that creature. In any case, these predictions highlight what we can actually do with the machine learning approach that we've developed. This approach corroborated the highly or fully aquatic habits we already knew about for ichthyosaurs and plesiosaurs. It found evidence for moderately aquatic or amphibious habits in the more ancient cousins of plesiosaurs and various stem reptiles, including mesosaurs, and it recovered at least highly aquatic habits for pleurosaurs and Spinosaurus. That said, again, I'd like to wait for the next round of results before we actually weigh in on the whole Spinosaurus debate. In particular, though, this gives us a clearer image for how some of the closest relatives of plesiosaurs lived their lives, regularly returning to land as needed. In doing so, this approach has already told us clearly just how aquatic these species were, and just how confident we can be about those claims. In this way, by combining old-fashioned measuring techniques with some steampunk naval statistics and modern machine learning, we found an approach that offers new clarity about the lifestyles of extinct marine reptiles. However, today we went over just a few select species, and the latest version of this dataset contains hundreds. So my next step will be to run the exact same machine learning model on all of the extinct species in the dataset to reconstruct the recurring evolution of aquatic habits in each of these groups more completely from start to finish. In doing so, we will add a great deal of ecological clarity and color to these remarkable creatures and have a more detailed picture of what life was really like during the time of the dinosaurs. Now, before we move on to questions, I'd like to thank a bunch of people who made this work absolutely possible. First of all, my collaborators who have believed in this project since its inception. Second, Aya Nawano, who I think is on Zoom right now, and many others for their hours of help with the computational stuff this project entails. All of the Peabody collection staff who are amazing, and dozens of others at museums across the world who helped me find and access specimens. Many, many people who provided critical feedback or training throughout this process, and of course, my funding sources. And in addition, I'd like to thank David Heiser and the Yale Peabody Museum for giving me the opportunity of speaking here today, Katrina Damrell for setting up this entire talk, and Bill Brailsford for organizing the technical aspects of this talk. And finally, thank you to my wonderful partner and family. I couldn't have done any of this without you guys. And of course, thank all of you for listening. I'll take any questions you might have. Thank you. In the front, did you have a question? Excellent.
To a layman, it would seem that
Yeah.
you will first distinguish aquatic from land-based
Yes.
tendencies by looking at breathing systems.
Oh, yes.
Looking at
Yeah. Yeah, no, great question. So breathing systems are one of those sets of features that's not very reliable for marine reptiles specifically because all of them breathe air. So in contrast to primarily aquatic creatures like fish that we think of as living in the water and breathing with gills, all reptiles, the ones on land and the ones in the water, and all mammals, the ones on land and in water, they all breathe air. So unfortunately, they're all similar in that way. And they've managed to, as much as they've been able to change their physiology and their anatomy and their behavior to handle life in the water, none, to my knowledge, no mammals or reptiles have like re-evolved the ability to breathe underwater anymore. And so, I mean, they can hold their breath for a little while, but those features were not the specific features because they wouldn't distinguish more aquatic from less aquatic ones. Great question. Yes, in the back. Oh, sorry.
Sorry, guys.
We're going to bring a microphone to you so the Zoom audience can hear the questions. Thanks for your patience.
It's thought nowadays that many marine reptiles, ancient ones, actually gave live birth.
Yeah.
So was that ever in consideration for the data set, or is that too
Yeah.
contentious a point or too vague a point in terms of all taxon?
Oh yeah, no, that's a great question. So live birth is interesting because in many groups of reptiles, it's people disagree about how much of a piece of evidence it is for aquatic habits. Lots of reptiles give live birth historically, and even now there are lizards that give live birth that live on land. But it is true that you kind of need to give live birth to be fully aquatic. Otherwise, you have to come on land at least some of the time, like sea turtles do to lay their eggs. And so live birth for example, plesiosaurs and ichthyosaurs and mosasaurs and their closest relatives has been used as evidence to say, hey, they're fully aquatic. They're never coming onto land. And if you see one in the middle of the land, a long time ago, it's probably going to die. But that is consistent with sort of how their skeleton is structured, which would not physically support their body weight on land. So all of those factors were taken into account when scoring modern and extinct species for just how aquatic they were. Yeah, good question. Kelsey?
Sorry, guys. Mic running.
Yes.
Okay. Would you?
Oh, that's okay. Oh, sorry. Did I?
Version of questions.
There we
Kelsey, do you
go.
Mind if we go first here? Okay.
Some of these animals spend some of the time in land
Yes.
predating as predators.
Yeah.
Spinosaurus is the opposite. It was mostly on water, if I understand, predate. So are you working on that?
Yeah. So I am working on those sorts of questions. One of the interesting things, and I'm glad you asked about Spinosaurus because interestingly, I mentioned it might be a false positive. Spinosaurus is an interesting case because theropod dinosaurs, the group that Spinosaurus is a part of, ancestrally have very long hands. And this run of the data set didn't have enough theropods to account for that fact, but the new run of the data set does. So I'll be really curious to see what sort of the next run of this model tells us about how aquatic Spinosaurus was. For those who don't know, there are sort of two main theories about how aquatic Spinosaurus was. That's very contentious. There are some theories that it was swimming underwater and actually diving deep to catch prey and things. And another theory is that it was kind of like a modern day heron, just snatching prey at the water's surface. And people disagree about that. And different lines of evidence suggest different things. So what's exciting is that this kind of approach can really take all of those modalities of data, all of those kinds of data, and bring them to bear on this question and see which one is most accurate. And we can do it. We can make an ROC curve for isotopic data. We can make an ROC curve for bone density data, and we can see which of those, sets of data is more reliable at predicting aquatic habits in modern species. And then we can make our decisions based on that. So I think this really has the potential to resolve some of these longstanding debates.
Awesome. And we've got Kelsey over here. And then after you, Kelsey, we'll hop over to the Zoom audience for a question.
Hi. First off, that was super cool. And I think this is like a major game changer in our field.
Oh my God, thank you so much. This is coming from an expert who just defended her PhD. So
You're
that's
too
great to
kind.
hear.
Well, while we're here, I wanted to ask, in your data set of living things, I
Mhm.
guess do you have a large sample of any sort of ontogenetic data, like babies
Oh,
and adults? And do
yeah.
you see any changes as things age?
Yeah.
Does that influence sort of the paleontological
Yeah,
guesses?
great question. So overall, we have a very large sample of juveniles, some neonates, or sort of like very late stage embryos or early stage just born, non-embryos, and adults. And so overall, we kind of have there's such an abundance of those things that the signal is stronger than the ontogenetic changes are able to disrupt. But there are and overall, I will say there is no correlation with, say, the size of the specimen we're looking at. There's no confounding factor about ontogeny overall across amniotes. However, within particular groups, there may be some confounding factors. For example, with the dolphins and whales in the data set, we have seen some differences in sort of how relative hand length changes with the age of the specimen. And actually, a former student is working on that right now who's on Zoom, I think. Hi, Jager, if you're there. And what's great is that the Smithsonian has assembled this massive data set of hundreds and hundreds of CT scans of dolphin flippers. And so we really have the amount of data, the sample size, to be able to look at intraspecific variation and ontogenetic change within individual species. So that's one of the projects that I've been involved with right now. To really ground truth some of these claims. But overall, for amniotes, it has not been a confounding factor. Great question.
Awesome. And David's got the mic back there for the Zoom question.
All right. Thanks, Caleb. There's a few questions here from Zoom that have to do with your modeling.
Yeah.
And I'm going to try to kind of summarize them into one, but
Absolutely.
they're mostly about how you chose which features. And so one of the questions is, how did you balance considering false positives versus false negatives?
Yeah.
Or does the model accuracy test both? And then the other related one is, why not consider all variables in the ROC analysis and weight them according to their usefulness?
Yeah, no, that's a great question. So, one of the things, this is a process where I get the reliability of a single feature for predicting aquatic habits at a time. Now, there are other machine learning approaches, for example, discriminant analysis, that you can use to combine all of the predictive features together into kind of like a prediction feature soup that you can use to predict aquatic habits all together. The reason I didn't employ those methods is because what you gain in accuracy, you lose in transparency. So even though those models might be slightly better at predicting aquatic habits, they take into account all of the data. they're only going to be marginally better, as at least it seems, from just relative hand length alone, since that model performs so much better than all the others. And then it's a lot less clear when you're looking at those graphs what the data actually mean. You know, what do these multidimensional values of various different predictors congealed together by the computer actually tell us? So what I really want with this approach is a method that can transparently tell researchers like me, oh, okay, hand length is a better indicator of aquatic habits than X feature. And those sort of multi-dimensional approaches don't do that quite as well. And then there was another question about why I chose the features I did. So why I chose the features I did was because I was really interested in limb evolution, and in particular in seeing if we could identify interdigital webbing in the fossil record. This is a soft tissue between the digits. If you think of something, you know, paddling in the water, or if you've ever put like a fin on to swim, it helps you swim. You swim a lot faster with them on. And some fossils actually preserve interdigital webbing in the fossil record. They're amazing, but the vast majority, of course, don't. It's a soft tissue. It only gets preserved in exceptional circumstances. And so I used these exact same methods initially to look for evidence of interdigital webbing in the fossil record. Could I predict that just with the bones? And it turns out you can't at all. All of the models looked like that finger bone model. They basically were not significantly better at predicting anything than you would be by just randomly guessing whether something was webbed or not. So my interest was really in limb evolution from the standpoint of webbing, in particular because I wanted to trace the evolutionary histories of these groups with a very large set of characters that I could measure for a lot of specimens. And it just so happens that the limbs are preserved for a lot of specimens. We have way more partial limb skeletons of fossils that are in this semi-aquatic range than we do absolutely beautifully, perfectly preserved skulls. So this was just a matter of sample size, that it provided the most data that I could really use to test this approach rigorously. And there was another question here, I believe. Oh, yeah.
Oh, thanks. Thank you. Yeah, I was wondering about vestigial limbs, if they have a place anywhere in your thinking, you know, which is in a sense a more advanced, further advanced evolutionary advancement.
Yeah, absolutely. Yeah. I mean, in some sense, a lot of the species that have flippers in a way kind of have vestigial limbs. So some of the mosasaurs, for example, in the data set have hind flippers that are like really, really small. And they're kind of like on their way out. They're like the human Y chromosome, like it's going to be gone eventually. And that's sort of like and so in that sense, those flippers were there, but they might still provide some function. And I didn't make any judgment calls about whether they were functional or not. One of the interesting cases and one of the limitations of this model is that the animal had to have limbs to be in the model. So some taxa are not touchable with this model. For example, sea snakes, I could not incorporate in this model because they don't have limbs. And so that's one feature that's worth noting, is like, to study the aquatic adaptations of sea snakes, you'll need a different set of data. You can still use ROC analysis like I did to find different sets of data that are better at predicting aquatic habits, but you wouldn't be able to use limb features because perhaps sadly, perhaps not, they don't have limbs. Yeah, thank you.
Great question.
Yes, Jacques.
Here.
Yeah.
Between forelimbs that are made to steer as opposed to propel. I mean, Stumpy downstairs obviously flew through the water,
right,
and used its hind limbs to steer.
Right.
But Red Cloud, the mosasaur there, was steering with its limbs and propelling itself with its tail.
Yeah, absolutely. One of the things that was so perplexing to me was that I didn't get a strong signal for that with the data that I collected, which is kind of remarkable because, so like Jacques said, and Jacques is one of the co-authors on this, like Jacques said, you know, animals are using their flippers for very different things. Dolphins are steering with their flippers in the water because they're propelling themselves primarily with their tails. But a sea turtle is swimming primarily with its flippers. Its tail is pathetic and tiny, you know. So so Jacques is asking, do we see any signal for how the animal is using its flippers in what the relative hand length or some of these other limb features are and how they differ between groups? And the answer, shockingly, is no. It seems that regardless of how an animal is using its flippers, whether it's steering with them or rowing with them or paddling with them or flying underwater with them like turtles do, regardless, its hand gets much longer relative to its lower arm. It's something that kind of like supervenes on that difference in a really crazy way. So it's pretty surprising, but for some reason we don't see it. That said, theoretically, this analysis with the right data could distinguish different predictive models that, support that are better at predicting how they use their flippers. Yeah, thank you.
It's a great question. I think we have a couple more here from the Zoom audience too.
Great. Yeah.
All right. Paul asks, given that early aquatic creatures' limbs, flippers/fins, evolved to have shorter arms and longer hands,
Yeah.
did the overall length of those limbs stay roughly the same?
Oh, yeah.
Were there different such evolutions for different species,
Yeah.
or does knowledge like that require further study?
Oh yeah, that's a great question. So did the hands get relatively longer or shorter? Overall, I think overall relative to the body, they got a lot shorter. Even the longest flippers relative to the body that I know of in the animal kingdom, the humpback whale today, or plesiosaurs, which have very, very long flippers relative to their bodies. I believe it's still shorter relative to the total length of the body than like our arms and legs are. So they have to reduce the length to some degree, or otherwise the torsional forces would just really hurt a lot and they'd get a lot of dislocated elbows and things. So they do reduce it, but they're modifying their limbs in a way so that it's more compact, so that they generate less drag, kind of so that they push up against the water in the wrong way less and in the right way more. And so that they generate more force pushing the animal forward as they swim. And oftentimes that involves the flippers, the arms, getting a little more compact over time. But different parts of the arms are getting bigger relative to the body. So like the hands of plesiosaurs are super, super long relative to the body as a whole. Much longer than our hands are relative to the body. And we do see that trend taking place also in dolphins, in sea turtles. If you look at the skeleton of any of these groups with flippers, it's like a tiny little stub of arm and then just a gigantic hand, which is kind of like crazy to picture. But that is the trend we see in all of these groups. I hope that answered the Zoom question, but if not, happy to clarify.
I think we have time for a couple more questions here and in Zoom. Does anybody have any more questions? No?
Yes.
Oh, let me hand you the mic.
Okay.
1 second. Come on up.
In evolutionary transitions,
Yeah.
from aquatic to land and from land to aquatic,
Yes.
do you identify same or similar ROC functions?
Oh, I see. So you're asking like, for example, so I was looking at, say, the limb to fin transition. How did terrestrial groups adapt to life in the water? But you're asking, do you see a similar trends and similar situations happening when animals that are on land, when animals that are in the water first adapted to life on land, when the first fish came onto land, for example? So that is, interestingly, it's a great question. This method theoretically could look at their aquatic habits as well. Some of them are in this analysis. So Tiktaalik is in this analysis. That's like a famous fossil that came out of a group at Yale Chicago that is kind of thought of as like the missing link between fish and animals with legs. But a lot of these fossils farther back, and to really do a careful analysis of that question, a lot of them farther back fall into the category that we were talking about with snakes before, where these creatures that evolved to come onto land were evolving limbs while they were doing it. So a lot of them don't actually have limbs that I would be able to measure with the same measurements that I was measuring things here. So, I assume things that for example, an animal has a humerus, that an animal has a radius and ulna and fingers that are measurable. And in order to make those measurements consistent across all of the specimens in the data set, they need to have those features. So in some of the first animals that were making their first wiggles onto land, if they didn't have a full suite of those features, I might not be able to include them in this data set. However, if someone developed another set of measurements that they could use similar to mine, but that could accommodate for the anatomy of those animals where the arm is not fully formed in the way that it is for us, then someone could repeat this process with all those measurements. Great question.
Since you mentioned fish,
Yeah.
how about coelacanth?
Oh yeah,
Um,
coelacanths
and,
are great.
And the question of the transition to breathing air as opposed
Oh
to
yeah.
gills.
Yeah, so coelacanths are interesting. I did show a coelacanth on one of my slides. I haven't asked my family permission to show their pictures for this entire time, so maybe I should go back. This is a better slide. There we go. Yeah, but, and there's a coelacanth on one of the early slides. Coelacanth is not in my analysis because this is like the perfect lead in from that question because it falls in that category. Our arms are homologous to the forefins of coelacanths, but they don't have all the same bones in their arms, particularly in their hands, that we do. So relative hand length I wouldn't be able to talk about relative hand length in the same breath for both of those groups of species, and I wouldn't be able to compare those measurements. So that would be something that this data set wouldn't be able to touch on, but a future data set could, looking at the fin to limb transition as these groups are first adapting to live on land. And interestingly, I mean, ancestrally, and Jacques can tell you a lot more about this, that coelacanths and the larger group that it descends from, that larger group probably ancestrally was breathing air to some degree. So it was more in a way the adaptations of the limbs and the spine that allowed them to really settle on land. And then even animals that go on land and have full limbs like amphibians today, they still don't have that special land-proof egg that allows them to lay their eggs anywhere without drying up instantly. Yeah. Thank you.
Great question. Oh, okay. A
Saw
couple more here.
Dalton had one.
Dalton was also in the Research Spotlight series, so.
Another
it's off on
expert.
YouTube, yeah.
Thank you. Caleb,
Yes.
this is an amazing talk, by the way,
Oh,
again.
thank you so much.
Do you have any thoughts on, given that it seems that it's such a reliable trend that as things become more and more aquatic, they relatively lengthen the hand and shorten the forelimb?
Yeah.
Do you have any sense on maybe like why that trend exists and why, for example, we don't see something that bucks that trend and has a really short hand and then tries to convert its arm into a forelimb? Do you think it's like constraints on making like these really tubular bones into a flat structure, or like maybe because there's a joint? Or,
that's a great question. Frankly, I mean, I find it very bizarre, and the short answer is no. I have no idea why, and I think it's a really interesting question. I don't know if there's something epigenetic about it, about how the forces are interacting with the bones as they develop over time, and how that gets compounded through generations. I don't know if there are changes in gene expression that are that maybe sort of developmentally the easiest thing to change with respect to gene expression is like pushing the expression boundary that specifies hand identity farther down. But frankly, I'm really not sure. From a functional perspective, I think it might be that increasing the length of the hand is just, and this is a bit of like a glib answer, but it's like evolutionarily easier, so to speak. But frankly, I think that's like a question for further studies from a bunch of different angles. I really don't know why they all, and like, without fail, all of the highly aquatic ones have longer hands than their terrestrial counterparts, which is bizarre. Now, there are some secondarily aquatic and highly aquatic species that have fairly short hands compared to others. So for example, manatees and dugongs don't have ridiculously long hands, but they still have much longer hands than their closest mammalian cousin. So this trend takes place within each group independently, which is bizarre. But yeah, in terms of why, I really don't know. And I think that's the cooler question. I'm not sure why they do that.
David, do we have any on Zoom?
Yeah, why don't we take this? Two people on Zoom were interested in whether you included any contemporary species in your analyses.
Oh,
And
yeah.
so I'll just read one question, which is, I think, most relevant. Were any extant avian dinosaurs, such as penguins, terrestrial non-flyers, and terrestrial flyers, such as hawks, included in your data set?
That is an excellent question. So one of the sort of, when I was designing this this whole approach, I said, I'm going to put one line in the sand. And for the same reason that I did with coelacanths and snakes, the homologies of the limb bones get a little weird when you get into birds. So like the measurements I would take wouldn't necessarily be consistent with birds and non-birds. So I excluded all bird forelimbs from this analysis necessarily, and all sort of flying forelimbs. So that goes for pterosaurs as well. Flying animals also, and there are very few of them, right? Just 3 groups of vertebrates fly, but they all lengthen their hands in the same way. And so it's kind of, I mean, in a way, if we were to include flying animals, it's any animal that's locomoting in 3 dimensions through a fluid medium, be it air or water, is increasing the relative lengths of the hands. But I excluded those taxa from this analysis. But interestingly, just anecdotally, measuring a few things and not including them in this analysis, shockingly, even penguins, so, you know, very aquatic birds, have much longer hand bones relative to the rest of the wing than flying birds do. And this is something that I've also seen in auks and some other groups. So I think there's a follow-up study here at some point to look at, you know, the ability at flying ability in general, but also aquatic habits in birds specifically.
And maybe the shoulder girdle?
Yeah. Yeah, absolutely. You know, I haven't even touched the shoulder girdle. So it's a whole other set of data to explore. And I know there are people looking at the girdle elements. And so the shoulder girdle is, these are your shoulder blades up here and your pelvis down here, the girdle elements. These are the bits that the parts of your body that hold your limbs to your spine, that sort of connect those pieces. And there are other people studying how the girdle elements change as this transition from land to water takes place, but I'm not one of them. I don't know too much about that, you know, so I think it's another you know, series of measurements, another set of data that someone should put through this machine learning pipeline and see what we get, see how reliable those characters are.
Thank you so much.
Yeah, thank you all. Appreciate it.